So I am thrilled to introduce a person who might be a new name to some of the people in the EHE community, but he is not new to EHE research. Jordan Driskill is here today to discuss work being done at the University of Texas Southwest Medical Center. And everyone, I'd like you to please welcome Jordan Driskill.
Hello, everyone. It's wonderful to finally be able to talk to you and to hear patient testimonials and also to hear from other researchers in the EHE field. So by way of introduction, my name is Jordan Driskill. I'm an MD PhD student here at the University of Texas Southwestern Medical Center in Dallas. And I work in the lab of DJ Pan. So our lab originally discovered the Hippo pathway and was one of the first to show that it's involved in cancer. And so with the wonderful work of Dr. Rubin and Dr. Antonescu, we became really interested in EHE and wanted to understand how the Hippo pathway and how the fusion protein itself are involved in driving cancer. And so today I'm really happy to show you two additional models that we've made in addition to the model that Brian just showed you. And so we also generated a genetically engineered mouse model, but we made it in a way that is different from Dr. Rubin's model. And we used this model to answer two key questions that I believe still exist in the field, or to show that to answer these questions in vivo. And we also generated an endothelial cell line that is transformed by TAS-CAMTA1, and that when transplanted into nude mice, forms progressive vascular tumors that are metastatic. And then I want to show you some data about a little bit of mechanism of how we believe this fusion protein is actually working by using these models. And then I want to just end by suggesting some novel therapeutic or some mechanisms for how some therapies that are already known may function and how some new ones may be used, which is what the ultimate question that we all want to answer. So to get into it, so the way we generated our transgenic mouse is that we essentially made TAS-CAMTA1 downstream of a tetracycline responsive element in the mouse genome. And so when we cross these mice to mice that express the tetracycline transactivator from an endothelial cell promoter, we essentially have an inducible and tunable way to turn on the TAS-CAMTA1 fusion protein in endothelial cells, okay? And so the way we did this experiment is that we essentially turned on TAS-CAMTA1 expression in endothelial cells in mice right after birth, okay? And then we followed these mice. And interestingly, these mice die within about 3 months, and penetrance is 100%. So whatever is happening very quickly in these mice. And if we actually dissect these mice and look for phenotypes, and what we observed is that these mice very rapidly, and many by about day 30 or day 40, the mice begin to show some signs of respiratory distress and lethargy. And when we dissected these mice, they often had fluid around their lungs. And so we dissected all of the organs, and what we observed is that we found the formation of vascular tumors within the blood vessels of the lung. And this is a very consistent phenotype and very widespread. And these tumors show many of the characteristic features of EHE and stain positive for many of the vascular endothelial cell markers, or EHE markers. And so we, like Dr. Rubin, argue that this means that the TAS-CAMTA1 fusion protein expression is sufficient to drive the formation of EHE. Now, the cool experiment we can do with our mouse model is we can essentially ask the question, so now that Dr. Rubin and that we've shown that this fusion protein is sufficient to drive the formation of EHE, what happens if we remove EHE? In other words, imagine you could go in the tumor and suddenly spontaneously take out all of the TAS-CAMTA1 and the TAS-CAMTA1 expression. What would happen to the tumor? Would the tumor keep growing? Would it stay the same, or would it go away? And so the way we did this experiment is that we aged our mice up to day 40. And the reason we did this is because we had observed that 100% of the mice had developed these widespread vascular tumors, which you can observe here, forming within the blood vessels of their lungs. You can see how widespread this is. And so then we fed the mice doxycycline water, which actually turns off the expression of TAS-CAMTA1 in these tumors. Okay. And what we observed is that the mice that were fed doxycycline, and which turned off TAS-CAMTA1, those mice actually lived a normal lifespan. Whereas the mice that had continued TAS-CAMTA1 expression died very quickly. And you can observe that in the blood vessels of the lungs of these mice, the tumors are gone. And so this really argues that continued TAS-CAMTA1 expression is required for the growth and progression of these tumors, and that inhibiting TAS-CAMTA1 will likely lead to death of the tumor because it seems to be required for its survival, at least in these early lesions. So next I want to show you our in vitro model. So there's an endothelial cell line known as MS1, and this was developed back in the late 1990s by Judah Folkman, who was one of the early researchers in the field of endothelial cells and tumor angiogenesis. So this is a non-transformed but immortalized endothelial cell line. So when we overexpress TAS-CAMTA1 in these endothelial cells, it allows them the ability to form colonies in soft agar, which is a feature that denotes transformation. And if we transplant these cells into nude mice, the control cells form this tiny 1 to 2 millimeter lesion that does not progress. Whereas the TAS-CAMTA1 expressing endothelial cells form this slowly growing progressive vascular tumor. And you can actually see that if you do histology on these tumors, the control cells form these benign hemangiomas that don't progress. But the TAS-CAMTA1 expressing endothelial cells form these large, solid, invasive vascular tumors with similar features as EHE. And these tumors can actually metastasize to the lung, which can be observed here. So we believe that this in vitro model is a very useful and simple way to study the function of TAS-CAMTA1 in endothelial cells. And clearly it shows some effects. It shows that TAS-CAMTA1 causes transformation in endothelial cells and also grants them metastasizing characteristics. So we performed a lot of fancy experiments like single-cell RNA sequencing on our mice, as well as RNA sequencing on the endothelial cell line to try to understand what is going on in these cells. And basically, we found, similar to what Dr. Rubin did, that there is a TAS gene transcription signature. So we very simply wanted to ask the question of what happens if you overexpress TAS, an activated form of TAS in endothelial cells? Do you also get EHE or these EHE-like tumors? So we did this experiment. We overexpressed TAS in these endothelial cells, and we saw that these mice died on a similar time course to TAS-CAMTA1 mice. And furthermore, we also saw the formation of these EHE-like vascular tumors in TAS mice. So this really argues that TAS-CAMTA1 is behaving like an activated form of TAS to drive formation of EHE. So then we wanted to ask the key experiment, which is, and others have talked about this, like Dr. Lamar and Dr. Rubin, which is that downstream of TAS, we know there's a transcription factor known as TEAD, okay? And so the question is, if you block this TAS-CAMTA1 TEAD interaction, can you actually rescue or prevent the formation of these tumors? In other words, is this a good therapeutic target? And so we crossed in this TEAD2 dominant negative. In other words, the endothelial cells that are expressing the TAS-CAMTA1 fusion protein are also expressing another protein that will block their ability to interact with TEAD. And here's what we observed, okay? So we saw, as we previously showed, that TAS-CAMTA1 causes lethality in these mice, okay? But if you overexpress this TEAD2 dominant negative to block that interaction, you actually rescue the lethality associated with TAS-CAMTA1. And furthermore, importantly, overexpressing or blocking the endogenous interaction between TAZ and YAP and TEAD2, which is represented by this green line, has no effect. So there are no abnormalities in these mice if you block this interaction under homeostatic conditions. This means that blocking the interaction between TAZ-CAMTA1 and TEAD will likely have limited toxicity, but have a huge effect on preventing the growth of these tumors. And you can see that when blocking the interaction between TAZ-CAMTA1 and TEAD, there are no tumors. They're totally gone. And our lab developed the first TAZ-YAP TEAD inhibitor, and we currently are working with many pharmaceutical companies to try and develop better inhibitors, as well as many other groups around the world. So next, I just want to show a couple of additional ideas that we've had from our data that may explain why some therapeutic treatments show some efficacy. Okay, so if we look at the transcriptomic signature of TAZ-CAMTA1 and the endothelial cell line, we actually observe overexpression of a gene signature known as VEGF-A. Okay, so VEGF-A is a protein that is known to activate growth in endothelial cells. And what we actually observe is that TAZ-CAMTA1 is upregulating the transcription of this growth factor in these endothelial cells. So they're sort of expressing a growth factor that's stimulating them potentially to grow more. And we can actually see that in these endothelial cells, when TAZ-CAMTA1 is expressed, that we're turning on genes associated with angiogenesis. Okay, angiogenesis is the developmental process by which endothelial cells form blood vessels and during development. So essentially, the idea here is that TAZ-CAMTA1 is sort of turning on these embryonic developmental gene programs in endothelial cells that are ultimately and aberrantly driving tumor growth. And if we utilize drugs that actually inhibit this VEGF activity like sorafenib, we can inhibit the growth of TAZ-CAMTA1. We can inhibit TAZ-CAMTA1 mediated transformation in these MS1 endothelial cells. And this inhibition is similar to the effect of what is observed when angiosarcoma cells are inhibited by sorafenib. And the significance of this is that multiple groups have argued that sorafenib shows some efficacy in patients. And so the one potential mechanism is that downstream of TAZ-CAMTA1, VEGF-A is actually turned on, which may lead to additional growth of the tumor. And then we also wanted to understand, sort of like Dr. Lamar, which is what are the possible negative regulators of TAZ-CAMTA1? And so we performed an unbiased mass spectrometry proteomic screen to ask what are all the proteins that are potentially binding to TAZ-CAMTA1. And what we observed, unsurprisingly perhaps, is that all the proteins of the Hippo pathway are binding to TAZ-CAMTA1. And these proteins, so they specifically bind to TAZ-CAMTA1 as well as to TAZ. And so the question is do any of these proteins that are known to inhibit TAZ, can they also inhibit TAZ-CAMTA1? And so we utilized a cell line in which LATS1-2, which are the canonical upstream hypophosphorylation kinases, when they're knocked out, because they normally negatively inhibit YAP-TAZ, whether these proteins are still functional. And so we observed that when you knock out LATS1-2, you actually increase the nuclear localization of TAZ-CAMTA1. Or if you knock out the hypophosphorylation sites of TAZ-CAMTA1, you actually increase their nuclear localization and increase their gene transcription signature. And so similar to what Dr. Kuskov showed, we also wanted to ask the opposite question, which is if you drive hippo signaling through some sort of drug, can you actually inhibit TAZ-CAMTA1's activity? So we utilized the same drug that she used, which is LAT-B, and we saw the same phenotype in endothelial cells, that when you treat cells with LAT-B, TAZ-CAMTA1 relocalizes to the cytoplasm. And the interesting thing about this is that there is an FDA-approved drug that acts similar to these sorts of LAT-B and blebistatin like she was showing, but at a much weaker, but has much less toxicity, and that is statins. So statins, which are one of the most commonly taken drugs in the world. And so simvastatin acts similarly to activate the Hippo pathway and to phosphorylate TAS-CAMTA1. And we observed that simvastatin treatment could actually inhibit the gene program of TAS-CAMTA1, and furthermore could inhibit its growth in soft agar. And the interesting thing about this is we did these experiments, and then we discovered independently that there was a study in the Journal of Clinical Oncology last year that said that our findings indicate a trend towards improved survival for patients with EHE who have received statins over the course of their disease. So we're currently very interested in continuing to follow this up further and see if what is the effect of statins on the Hippo pathway and on TAS-CAMTA1 itself. Anyway, so I'm really happy to show you these two new models to argue that TAS-CAMTA1 acts like an activated TAS, and that blocking the TAS-CAMTA1 TEAD interaction will likely be highly effective, and that this interaction is required in our mouse model. And that therapies that target VEGF-A or Hippo signaling can inhibit TAS-CAMTA1 mediated activity in endothelial cells in vitro. And so this project was done in our wonderful lab in Dallas. And we had spectacular assistance from Dr. Michael Dellinger, who is a vascular anomalies expert, and Dr. Dinesh Rakeja at Children's Hospital here in Dallas, who helped us with pathology. And our funding for this project was provided by HHMI and the NIH. So at this point, I'm happy to answer any questions that you may have.
Well, first, let me just comment, that's an amazing amount of work on a lot of different fronts. Congrats on that team being so wonderful. You don't see that much coming out of a lab at that level on so many different fronts very often. So very nice work.
Thank you.
Handful of questions here. So
okay.
Some of the data you showed is that after you were able to shut down the system, basically the cancer disappeared. Do you have any feeling for the mechanism by which sort of existing tumors went away? Is this a starvation event? Is this an immune system? Any ideas what's going on there?
Yeah, it's a great question. So what we observed in our so we looked at a lot of different markers. So the big question is, what is TAS-CAMTA1 actually doing in these endothelial cells? And the most obvious thing we saw is that TAS-CAMTA1 is really driving proliferation. And so when you turn off TAS-CAMTA1, that proliferation goes away. The problem with our model is that we don't have big, beautiful tumors like Dr. Rubin has, and our mice die very quickly. And so this is something that we are trying to answer with our xenotransplantation cell line in our nude mice. But if I had to guess, it would basically be that because you're turning off the proliferation, it's likely that programs that are turning on cell death or immune system or a variety of different pathways are helping clear these tumors that are developing within these vessels. But we don't know specifically what's happening, and we would like to, of course.
Got it. So we got a question here about statins and asking basically, what specific statin helped? And you mentioned one in the data. Have you looked at any others? What's your feel for at this point about the nature of statins that might be useful?
Yeah, so it's the literature on statins is actually very interesting. So simvastatin on these MS1 endothelial cells is extremely sensitive. So we can inhibit transformation of these endothelial cells with a very low concentration. Okay, we've tried simvastatin and pravastatin, and both of them work to inhibit TAS-CAMTA1 mediated transformation in endothelial cells. But I would say that pravastatin must be used at a much higher concentration. And for some reason, actually, people have known, and if you look in the literature, simvastatin has some effect on endothelial cells in culture that causes cell death that is independent of this classic YAP-TAZ hippo signaling pathway. And people don't understand what that may be. But it's really interesting to think about in the terms of vascular tumors, if that pathway can actually be harnessed, and why that is different from other statins like pravastatin. The study that I referenced from JCO, they just looked at globally patients on statin treatment. So they didn't look at like simvastatin, pravastatin, or anything like that. And in the hippo field, people have used pretty much any statin they can get their hands on, such as atorvastatin and others. But I think just because I see that simvastatin is so much more potent than pravastatin, there's some question of like why some are better than others, and whether that actually matters in patients or in vivo, because we didn't show any of that data, or whether this drug can actually work in vivo.
Gotcha. Thank you. Another question here. Dr. Rubin showed data in his model that EHE predominantly developed in liver and peritoneum and less commonly in lung and pleura. And you're showing predominantly in lung and pleura. Can you comment at all about on the potential biological differences in the models to explain that?
Yeah, absolutely. So Brian's model is very nice in that he can turn on, so we're both using the same sort of marker, which is CDH5. He's turning it on with CRE and we're turning it on with a TTA. But the problem with our model is that it is possible that we're actually turning it on higher. So we're expressing it more in the lung as opposed to in other tissues, which may be why we're specifically seeing the tumors form in the lung. And the other caveat I have to put here is that Brian's model takes some time for these tumors to develop, and our mice are dying very quickly. So it may be that the tumors would form in the bone and the soft tissue and the lung if they lived long enough. But we have not observed any sorts of lesions developing in these other locations. We have observed lesions developing, for instance, in the skin and in fat and soft tissue, but this is not as consistent as the phenotype in the lung, which is why we focused on the lung.
Got it. Thank you. Specific question about a specific drug here. It's one I'm not familiar with, so I may botch the pronunciation. But have you tried propranolol? Are you familiar with?
Propranolol. So, this is a really interesting question because propranolol is known to inhibit the growth of hemangiomas, and the other thing is that we know that propranolol works on G protein coupled receptors, which also act on YAP-TAZ. So we haven't specifically tested propranolol or other G protein coupled, agonist or antagonist, and I think that would be really interesting. Future study, to ask the question of whether these GPCR modulating drugs could also show some efficacy, since we have shown that there's some activity through hippo signaling, and Dr. Lamar's work, hippo independent mechanisms that may control TAZ-CAMTA1. These are all great ideas, and I think our model will be very useful to answer these questions.
With your in vitro model, you showed metastasis to lung. Have you characterized those metastasized cells to see if they are looking like, in any meaningful ways, what we're seeing with metastasis in human patients?
So I am not a pathologist, even though I would like to be. So I don't want to speak too affirmatively about this. What I would say is, based on some of the images that people showed earlier, I mean, they look pretty good. They form these lesions all over the lungs that have these sort of characteristic, like I don't know how to describe it. They're like big tumors that are very circularized, sort of as other people showed. So I think it's at least a good model to show that this fusion protein really can drive metastasis and growth in endothelial cells, and and Brian's work really shows that this fusion protein alone is sufficient. And so I think that all of our work really points to the fact that we need to really think about targeting this fusion, and that will be highly effective at preventing metastasis and growth. And so I'm really excited about that angle.
Excellent. Well, lots of work to do in lots of different directions. Thank you so much for your time. That's the questions I've got, and we look forward to hearing more from you in the future on all of these fronts.
Hopefully in person.
That would be great for so many reasons.